everyone, and welcome to this talk. I am Mark Meijerink, and today I would like to show you some of the work we did at Utrecht University. And this work is on using liquid phase transmission electron microscopy with the protochips Poseidon holder to study catalyst particle growth at the individual nanoparticle level in a liquid phase, of course. And we will be focusing on gold nanoparticles in this work. But first, I would like to discuss a little bit of why this is important for us. So heterogeneous catalysts are usually metal or metal oxide nanoparticles of less than about 10 nanometers. But because these particles are so incredibly small, they have a very large thermodynamic driving force to grow. And this is unwanted because catalysts usually catalyze the reaction only at the surface. And this particle growth also makes the surface area decrease a lot. To prevent this, particles are usually anchored on a very high surface area material called a support, like this here. But even then, what we often see that particle growth is still significant. This growth is usually happens through one of two mechanisms. The first one being particle migration and coalescence, shown here on the left. And in this, smaller particles move around over the surface or sometimes through the fluid. And then when they meet, they merge together to form one larger particle, causing particle growth. The other one is called Ostwald ripening. And here the particles themselves stay in place, but individual monomers can detach from the particle, then move either over the surface or through the fluid, and then to another particle where they then can reattach. And this movement is usually assumed to go only from smaller particles to larger particles. And this causes the larger particles to grow even bigger and the smaller particles to shrink and eventually disappear. Unfortunately for catalysts, this particle growth is very difficult to study because the particles are so incredibly small. One technique that you can use, however, to observe this particle growth is transmission electron microscopy. But the challenge is that this technique can only observe materials in a very high vacuum, which means that you cannot actually use liquid in the microscope because it will evaporate and ruin the vacuum very quickly. So when studying this particle growth, what is usually done is you take your sample beforehand, put it in the microscope, and then you get something that looks a bit like this, with the very tiny dots being small particles. Then you do your catalysis, take out the sample afterwards, and then look at it again. And then you might, for example, see something that looks a bit like this, with now very large white particles having grown. But the question still remains to some extent, what is actually happening here in between? So one way that you could actually look at this is by using a liquid phase transmission electron microscopy. And the idea basically is here to sandwich your sample and some liquid between two membranes. This keeps the liquid separate from the vacuum, but also still keeps the overall system small enough, thin enough for the electrons to pass through and actually still image your sample. So there's multiple ways of doing this, but the approach we used here is by sandwiching our sample and liquid between two silicon chips with a very thin silicon nitride membrane, as you can see here. And then at some places the silicon is etched away to only leave the silicon nitride membranes. And then this part is thin enough for the electron beam to pass through. The way you would do this in our case is by depositing your catalyst on one of the chips, and then some of it will also attach to the places where the window is. And then you take another chip, and put your liquid and a sample between these two chips in a specialized holder. One of the chips usually has also some pillars of a certain height that are usually referred to as spacers. And these are important to keep the two chips separate to make sure that they don't attach to each other and basically push out all the liquids. There's a few different considerations that you would have here to optimize your resolution. And these are, in our case, are mostly the spacer height, the overall area of the window, so the length and width of this window, and then also the window thickness. So the spacer height, because it, for the most part, determines the thickness of your liquid, is important because then the thicker your liquid is, the more liquid reservoir you have. And for example, the more reactant you would have in your solution, if that's important. But also a thicker liquid means that the resolution will be lower. Another important aspect is the area, overall area of the window, because a higher area means that you have more sample visible and you can look at more of your sample, which can give you better statistics. But a problem is that because there's a big pressure difference between the inside and the outside of the liquid cell, the windows actually start to bulge a bit, locally increasing your liquid thickness again. And this will then again lower your resolution. Another aspect is that the window thickness itself can be varied. And a higher thickness means that the window is actually stiffer and will bulge less. But then, of course, you have you have a thicker silicon nitride layer, and this will also decrease your resolution again. A second aspect might be that if you have a very aggressive medium, you might actually want thicker windows, because then they will last longer in your experiments. Because if you have a very aggressive liquid, you might actually slowly etch away your windows during the experiment. And this might also be something that you would want to consider, but in our case, is not that much of a problem. So we are looking at gold nanoparticles, which are very good catalysts for biomass to chemical conversion, but they are very well known for growing extremely fast during catalysis. And this is especially the case in the presence of chloride ions, which are also unfortunately a very common contaminant in biomass. And this is why we would really like to study the growth of gold nanoparticles. So the main reason they accelerate growth is because gold can stabilize, because the chloride can stabilize ionic gold species. And then according to this reaction, you will dissolve the gold of your gold nanoparticles very quickly. And then the gold can move through the solution and to another particle and then reattach there by the reverse of this reaction. And this also is then results in some form of Ostwald ripening. So then we would really like to study this in situ while it's happening. And that is exactly what is shown in what we did, and the result of which we are showing in this movie. So this is a gold supported on titania catalyst, with the gold nanoparticles being the very the small dark dots. And this gold on titania catalyst is in contact with water with 10 millimoles of sodium chloride dissolved in it. And this movie is about 1 hour in real time. And then we will be looking at how the particles change. And I would like to especially focus on the particles with the 3 particles with the arrows, starting with this particle with the green arrow. So as you can see, this particle, like all the other particles, stays in place, and in this one very slowly just grows over time, as you would expect from Ostwald ripening. But then when we look at this particle with the red arrow, Again, this particle also slowly grows over time and still stays in place, again, as you would expect from Ostwald ripening. But then at some point, very quickly, it completely disappears. And then you might have already seen it as well, but this particle, which is initially the biggest particle in the system, again stays in place, but even this one disappears very fast at some point. So we also measured the size of the particles in the movie over time, and then that is shown in this graph over here, for a few particles. Each line in this graph corresponds to the size evolution of one individual particle. And so we saw three main distinct behaviors. The first one being the behavior of a particle with the green line shown here, in which the particle just slowly increases in size over time. Then the second type of behavior is the particles with the red lines. So here the particles either stay the same size roughly, or even sometimes grow slowly over time. But then at some point, almost immediately, the particle just completely disappears, completely dissolving very likely. And then a third behavior is somewhat unexpected, which is the blue lines, in which the particle more or less stays the same size over the entire experiment. And this is somewhat unexpected because Ostwald ripening theory usually predicts that the particle will either grow or shrink, but not stay the same size. But what is most interesting, what you might already have seen in this graph and in the movie, is that the particle size actually does not seem to affect the type of behavior it displays very much, which is very much in contrast to what classical Ostwald ripening theory predicts. So then the question might be, are we actually observing Ostwald ripening? Which is a very good question. But for that, we also measured the particle size distribution of a lot of the particles, both in situ and ex situ. And what you can then see is the particle size distributions actually show particle growth very much akin to the particle growth you would see during regular Ostwald ripening experiments in practice. And both the behavior that we saw inside the microscope and what we saw in lab scale experiments match very well, actually. So we are indeed observing Ostwald ripening, typical Ostwald ripening that you would see in other experiments as well. And one other thing that we saw is we could also, from these particle size distributions, extract the specific surface area, or the surface area per gram, which is very important for catalysis because the catalysis happens on the surface, so you want to keep this specific surface area as high as possible. And then we could also fit this observed behavior to a typical rate equation that is shown over here. Which is usually the way that specific surface area decrease is described in actual catalysis. And then you can see that the fit we saw with sodium chloride actually matches pretty well with the experimental behavior. And also for water, it fits pretty well, but is, as you would expect, a lot slower than when sodium chloride is present. And this also shows that this can be very useful for a better understanding of how Ostwald ripening actually happens during actual experiments. So to quickly summarize what I've shown here. So we have shown that liquid phase transmission electron microscopy can provide direct evidence for liquid phase Ostwald ripening on the catalyst we use here, namely gold nanoparticles supported on titanium dioxide. The behavior of the entire ensemble of particles matches very well with ex situ results and also what is seen in other people's experiments. But at the nanoscale level, the mechanism is very different from actually classical Ostwald ripening theory. And which we can see here is that growing particles do more or less behave like classical theory, just growing very slowly over time. But particle shrinkage is very quick and very fast, and seems to be more of a stochastic process. And what is also interesting that we saw a very significant fraction of particles, almost 1/5 of the particles, does not seem to change in size very much, which is also somewhat unexpected. But most importantly, we saw that the particle size actually does not seem to matter much for the behavior it displays in these experiments. We also have we also discussed this in a bit more detail in the paper we published in 2019. That is shown over here and will also be in the video description. And with that, I would like to thank also the following people for their help in this work, and ERC and NWO for the funding. And of course, thank you for watching.